In this lab, we are going to be studying the structures of carbohydrates. In our bodies, carbohydrates have many functions, including providing an important source of energy for our bodies. Monosaccharides are simple carbohydrates, meaning there's one unit. They are either polyhydroxylated aldehydes and ketones. So an aldose is a monosaccharide that contains an aldehyde as a functional group in its structure. So for example, D-glucose, which is the primary form of energy in most biological systems, is an example of an aldose. So it has an aldehyde group at carbon number 1, and then attached to the other carbons 2, 3, 4, 5, and 6, are hydroxyl groups attached to it. D-fructose is an example of a ketose. So a ketose is a ketone-containing carbohydrate. In this case, the ketone group is at carbon number 2, and then all of the rest of the carbons have hydroxyl groups attached to it. Monosaccharides are able to form cyclic structures. What happens is, in solution, at least for the monosaccharides that we are going to study, the hydroxyl group that is attached to carbon number 5 of the monosaccharide will react with either the ketone or aldehyde group. So in this case, we have D-glucose. Here's the aldehyde group. Here is the hydroxyl group. So the hydroxyl group will react with carbon number 1, that double bonded oxygen, to give us a cyclic structure. And so after it cyclizes, it forms a hemiacetal structure in which we have this carbon, oxygen, carbon, hydroxyl group structure. So this is a hemiacetal. So what you can see is that the original aldehyde group in this case that was present in the linear structure, that is now contained within the hemiacetal group in the cyclic structure. And this carbon within the hemiacetal group, we call this the anomeric carbon. So aldoses, at least 6 carbon aldoses, they will form a 6-membered cyclic structure. Now ketoses, a very similar type of reaction will happen. Again, the hydroxyl group at carbon number 5 will react with the ketone group, so that double bonded oxygen, to form the hemiacetal structure, forming a what we call the anomeric carbon. And with ketoses, they will form a 5-membered ring. Now with monosaccharides, this cyclization is a reversible process, so they are freely able to rotate between the linear form and the cyclic form. We are now going to move on to disaccharides, which are carbohydrates that are composed of 2 monosaccharides joined together. And when they join together, they form what's called a glycosidic bond to form the disaccharide. So the way this works is, here we have 2 monosaccharides. This shown here is D-galactose and D-glucose. So there are 2 different types of monosaccharides that exist. In the reaction to form a disaccharide, the anomeric carbon of the first carbohydrate, so that of D-galactose, will react with the hydroxyl group of another sugar. So in this case, it's carbon number 4 of D-glucose. And when this happens, a molecule of water will be removed. So the hydroxyl group from carbon 1, or the anomeric carbon, and the hydrogen from the second hydroxyl group, to form this O-glycosidic bond. Okay, so this together is a condensation reaction because a molecule of water was removed to form that linkage. So other examples of disaccharides out there are lactose. So here we have the anomeric carbon of this sugar, so this is carbon number 1, in a glycosidic bond with carbon number 4 of the second sugar. Here we have maltose, which is a very similar type of arrangement where the anomeric carbon is in a glycosidic bond with carbon number 4 of the second sugar. And then trehalose is a bit different. So both anomeric carbons are in a glycosidic bond. And then here, this is sucrose. So we have an aldehyde and a ketose that are in a glycosidic bond. And so again, both anomeric carbons are in the glycosidic bond. Polysaccharides are carbohydrates that are composed of many, that is more than 3 monosaccharides. An example of a polysaccharide in our polysaccharide is starch. There is a specific type of starch called amylose that is able to actually interact with iodine, and when it does, it forms a deep blue color. In this lab, what we will do is perform 4 different chemical tests, which will tell us the structural properties of various sugars. So in the Benedict's test, this will identify monosaccharides or other carbohydrates that have a free aldehyde or hemiacetyl group. That is, they have a free anomeric carbon. These are called reducing sugars because they will actually reduce copper 2+ to copper 1+. We will do the Barfoot's test, and this is a test that will distinguish monosaccharides from other reducing sugars because monosaccharides will react faster with this reagent. There is Siliwanos reagent, which will distinguish ketoses from aldoses because ketoses will react much faster compared to aldoses. And then iodine, which is going to detect polysaccharides by forming a deep blue, black to red purple color with it. So first, let's talk about Benedict's test. Benedict's test is a test that is going to test a reducing sugar. So what do I mean about a reducing sugar? Well, a reducing sugar is going to have a free anomeric carbon. And so, shown here is D-glucose. Here shown here at carbon number 1, that is the anomeric carbon. And notice that the hydroxyl group is not bonded to anything. Because of that, it is able to linearize, and then the linear form of the sugar is able to react with copper 2+ and reduce copper 2+ to copper 1+, and ultimately forms copper oxide, which is a red precipitate. So in this case, D-glucose will test positive on a Benedict's test, and it will turn red because it has a free anomeric carbon. Therefore, it is a reducing sugar, meaning it can reduce copper. Now, some disaccharides will also test positive as well, as long as it has that free anomeric carbon. Here is lactose. You can see that the hydroxyl group at the free anomeric carbon is not bonded to anything. So again, it will react with copper or Benedict's reagent to form that red precipitate. However, you can see that sucrose would actually test negative because both carbons are bonded together. So they are in an O-glycosidic bond. There is no free hydroxyl group at that anomeric carbon to be able to linearize and be able to react with copper. Barfoot's test is a very similar type of test, except the difference is that it distinguishes between monosaccharides and disaccharides. So it's actually a variant of Benedict's test where we're still looking for copper 2+ to become reduced to copper, which is why a positive test would have a very similar result of a red precipitate. And it's also going to react with free anomeric carbons. What makes Barfoot's test a bit different than the Benedict's test is that the monosaccharides are going to react much faster than disaccharides. So a monosaccharide such as glucose or fructose should give a positive result within 5 minutes, whereas a disaccharide should give a negative result, and it'll take much longer for a positive result to happen. Now, I should mention that if the sample does not contain any sugars with a free anomeric carbon, it should give a negative result. Next is Seliwanoff's test. This detects the presence of ketosis in the sample. Just a reminder that for this experiment, we can identify ketosis and aldoses in the cyclic structure because ketosis will be a 5-membered ring, whereas aldoses will be a 6-membered ring. So if a ketose is present, then when it reacts with Seliwanoff's reagent, it will give a cherry red color that will happen pretty rapidly. So they'll look something like this, whereas aldoses, it will react much slower with the reagent and it'll produce a light pink color. And other materials will not give a color change. And then lastly, the iodine test will test for polysaccharide. This is because polysaccharides, or some polysaccharides, they will form a helical structure, which will then bind the iodine reagent and cause the solution to stain a very deep blue color. And so these results are very, very obvious and very immediate.